All right, we've got our second lecture on mutation. And the first one, we basically introduced a couple of ideas, general ideas about mutation, mutation rate, and we talked about transition versus transversional mutations. And at the end of that, I said, okay, let's think about now what happens if we have a transversional mutation or a transitional mutation. What can it cause? These are called substitutional mutations because we're just going to change one base, right? We're not going to delete a base or add a base. We're simply going to change the base. And there could be multiple outcomes, but three we're going to look at are what are known as silent, nonsense, and missense. Okay? A silent mutation is one where we get no change in the actual protein, sequence of the protein. Okay? Very simply, here's a case where you've got, because of wobble, right? You have the ability in this particular case to change any of these three last base into any one of the four possible bases that you would have. And that will still give you serine. So if you mutate the DNA at this position, it has no effect on the outcome of the protein. The protein will still be met his serine, arginine, proline, right? So it can be, and very often is, a change in the third position that causes no change in the amino acid coded for. This occurs because remember that term we talked about, the genetic code, back when we did the translational exercise, is that there's degeneracy of the code, right? We said it was unambiguous going down. There's no question that UCA will give you serine, okay, as much as UCU will give you serine. However, there's degeneracy because serine can be produced by any of these, in fact, more than just these changes, right? So there's degeneracy of the code. That makes it silent. It's not seen. A little mimic that I would have is something else, maybe something like this. The cat saw the dog hit the can or something, and we change it to little g. Okay, had no effect on the way we would read it live, but it has no real effect. Now there are some ways that this can be detected, but for us, as far as the phenotypic production, okay, that occurs is not going to be changed, right? So it doesn't have to be third position. There are several times when it can be first position that results in no change, and there's even some other ways we can change. But a silent mutation, a point mutation is what we're talking about here. Points, single nucleotides that doesn't have any effect on the amino acid sequence is clearly a silent mutation. Okay, this reason this is in blue over here, a change in the genotype caused no change in the phenotype. When you get into more complicated genetics and get into population genetics, there's what's known as silent mutations that even though they change the amino acid, they don't change the phenotype. Okay, sounds impossible, but it's not, and therefore they're considered silent, even though they're formed. So once again, just like we talked about, at a nucleotide level, it might be a codominant mode of inheritance, but at the phenotypic level, it might show up as a dominant mode of inheritance. Well, it's the same idea here. Now, for us, for the most part, and things that'll be consistent with any genetics text you see, is that it's a change in any codon that results in no change in the amino acid, very often associated with the third codon. Okay. All right. Well, then we move to ones that do have effects. Missense is where we do change the amino acid sequence. So we'll just start out with one that looks like that. It's got our MET here, histine, serine, arginine, proline. Okay. We make a single base change, in this case in the first position. All right. And that changes what we end up putting down here. Okay. And so if we take our little silly kind of limerick here, the cat saw the dog. Okay. If we make some little change in there, we might change it to the bat saw the dog. Okay, now in that case, it's still functional, but it probably gives a different protein. Certainly, bats don't see very well, certainly not as well as cats. They use, their other radar senses their sonar senses to detect things, but they can still see. So it does make sense, but it's a different model, and that's exactly what happens here. And again, depending on where this occurs and which amino acid is being changed, it can have essentially no effect on the phenotype, or change of the function of the protein, or it can have a dramatic effect all the way up to lethal if it's the wrong type of change, right? A good example of that is a simple one where we have this long amino acid here series, and of course we're reading them as codons, and we've got proline, glutamic acid, and we mutate a second position, which almost always causes a change in the amino acid. And this time it's valine. And it turns out that causes a major effect. This is one of the primary ways which sickle cell anemia, sickle cell trait, is the result of a single base change that is a missense change. It's in the beta chain, it's in one amino acid, and it causes these red blood cells not to have full form, but in fact to sickle, right? To cause them to have a collapse state, which causes all sorts of secondary problems, such as they hang up instead of rolling through the capillary beds as they should. They'll hang up and they cause problems therefore in the kidneys and livers and all sorts of other places where we have lots of capillary beds. And this leads to what's known as sickle cell anemia, and it's in your joints and elsewhere. Okay, so they can go from no effect. There is an effect in that they change the amino acid. Phenotypically, it can be no effect all the way to lethality and everything in between, just depending on where exactly and which amino acids are being changed here. Okay, all right. Then we have what's known as a nonsense mutation. A nonsense is where some codon gets changed to a stop codon. Okay, so we'll go back to our little model here, right? We're going to work on this middle one. This one is producing a serine. All right, let's make a single change. This time we're going to change the second position, and we're going to change it to a G. Well, you know from what you should know for the exams is that UGA is a stop code. It's a term code, right? So now we have a protein that is much shortened. Okay, this one's 3 amino acids, but wherever it is down the protein, it's now shorter than it would be. So instead of the cat saw the dog, we just say the cat saw the. Well, that's not a complete sentence. It's not even makes sense. And very often that's exactly what happens to the protein. If you don't have a full-length protein, then you don't have the ability for that protein to fold correctly, and therefore its function will change. Just a couple of models of where that might be. We see in cystic fibrosis, it's one of the most common genetic diseases worldwide. There is a protein that is part of a transmembrane protein that results in essentially the sticky material that gets pushed out of the cell. It's a misformed protein, and it's caused by this nonsense mutation, an early stop code. Okay, Duchenne's muscular dystrophy, which it has, and again, it's 15% or so, or some proportion of it. And there are multiple ways you can get both of these diseases, obviously, but this one, they have this low level of dystrophin protein, which is common in everybody with Duchenne, but for different reasons. But in one particular case, they have the protein, but there's a premature stop. And so they don't end up with the right amount. The functional part of the protein doesn't work. Dystrophin is really important for muscle tone and for structure of skeletal muscles in particular. So these individuals, once they reach their teens and early 20s, start to generate their muscle capability and eventually become where they can't function, can't move very well at all, and usually it is a lethal condition. Cystic fibrosis, if it's not treated, it will also be a lethal condition in people reaching maybe their 30s or so because they can't expel the materials from their lungs and that sort of thing. Okay? Now, this is one of the interesting areas. This is where some pharmaceutical systems have come into play. Okay? If there's one which is trying to figure out how to, in fact, solve some of these problems. And one of them is that they realize that there's a stop codon. The message is the right length and it's there, but you've got a stop codon that's in the middle, right? You've got this not making a full length protein, and therefore it's an incomplete and non-functional protein that's being made. Well, some drug companies have figured out ways to actually go in and using medications, they're able to prevent it from stopping at this first point. Okay, there's a translation that kind of moves on past it. Now imagine what's really got to be done here. You have to understand the message that you're looking at. You have to understand the mutation that's there. You have to think about what unique structures of the particular nucleosome that I'm going to have that's going to, excuse me, ribosome I'm going to have that's actually going to make this function, right? And how can I go in and interact with that ribosome so that in that whole complex that will actually allow the transitional process to move forward. And there's been some success in doing this. And then once you do that, you have a functional protein, and the problem, the cystic fibrosis problem in this case, is helped, right, in some ways. And there's a lot of this stuff experimentation going on, but it takes some real molecular level and cellular level understanding to try to how to put all this together and make it function in a way that will work. And again, this is not going to cure everybody because it's not the same way that everybody gets it. But for those large percentage of individuals that have it based on this mutational form that puts in a stop code, it would potentially work or certainly make their lives better. All right. Well, that looks at these three groups of substitutions. Now let's talk about, as we've done before, we went to the bigger things, which are deletions or insertions, right? And the two we're going to talk about, your book covers several that you should look at, but we're going to talk about frame shift mutations and what's known as in-frame mutations. Okay, so let's look at frame shifts first. Frame shifts are a gain of loss of one or two nucleotides, okay, that result in a change in the reading frame. Okay, so let's think about our same little model that we've had here, right? We've got a little message. We're producing the appropriate amino acid sequence that's here. All right, but let's do something. Let's do a deletion. So we're going to take this U right here and we're going to remove it and we're going to delete it out. Okay. Well, remember that once we go in here and we initiate the process using the small subunit and we frame the first, whatever the first codon is, with, in this case, MET, which it would be normally, okay? It's three letters after that. So it's a dumb code. The system has no way of knowing that it took something out or put something in. It simply takes the next three of these nucleotides found in the RNA, reads those as the next word, grabs the next one and the next one. So that's exactly what's happening here. The U's gone. So instead of reading UCA, it grabs the C, the A, and then grabs the C from what should be the next codon, the first letter of the next codon, but it's not. It's a dumb system. It doesn't know any better. It just grabs those next three, reads it as CAC, and it changes the outcome, okay? And you end up with a different form. So you might see, and what it often does then is everything after that is going to be messed up, all right? It's going to be unintelligible to the protein structure that we're trying to deal with here. We've changed it for. And one of the things that we did in lab was we made you look through some DNA and we asked you how many places do you see if you were to take that and make a message out of it, would you see a stop code, right? One of the things that we worked on. Well, if you don't know what the framing is, there are lots of places that could be a stop code. And that's one of the secondary effects of these frame shifts. When you have a frame shift, very often what's going to happen is that somewhere down the line, you're going to grab three of these nucleotides that represent a term code. It's not supposed to be there. So you not only get a misplaced and then a problem for the rest of the protein as far as the amino acids that are supposed to be there, you also often end up with a shortened protein. Okay, and this can occur from either losing a nucleotide or two or gaining a nucleotide or two. Okay, it doesn't just have to be one, but it's usually symbolized by one. Okay, probably one of the best examples I can give you that is Crohn's disease. It's a huge issue that affects a large number of people in Europe and North America. It seems to be one that's from that region tend to be more affected. It's obviously an inflammatory bowel disease. You probably know someone that has it. Very discomforting. This one involves an insertion, so a gain, right? And it's in a particular gene. And this is not every Crohn's individual, but this is one of the ways you can have this mutation. And what it involves is insertion. Okay, and now the system's still a doofus system in some way, okay, as I call my dog. We read codons as codons, so that's three letters, AAC, and then we should be picking up ACU, but we're not. We're picking up a CAC now, and then we're shifting this UGA over. Uh-oh, think about that, right? Think about what we just did. We picked up a CAC, and now instead of having GAG, we have UGA, and I think most of us recognize that UGA is a stop codon, right? So this frame shift, okay, by Adding in one actually ends up ultimately being a nonsense, what we would call a nonsense mutation, right? It's equivalent to that. It's not technically a nonsense is simply a substitution that somehow would have made UGA out of one of these codons. But here it effectively ends up like that, right? We've end up with a stop codon here because we've inserted. Now in this case, I can't think of a pharmaceutical way to go in and treat this because you are reading these codons. You're not stopping and then all the other codons are in the right place like it would be in a true nonsense. Here you've got a problem that you're just, you're completely messed up from the rest of it. All right, well, that's a frame shift. Again, you can gain or lose one or two nucleotides and affect the protein itself. Okay? And then the last one of these we want to talk about is an inframe mutation, and that's exactly what it says. Inframe, think about frame. Frame is a codon. Codons are 3 nucleotides, so gaining or losing 3. Okay, just gaining or losing 3. So in this case, we're just going to drop out this UCA, this serine, and it's going to pull the arginine over as the next one. And so it would be something like this, okay? The cat saw the hit the can. And the point of that, trying to make that, well, that's not really a sentence. Well, no, this is not really probably a protein. Now, if it's some protein that's just out here, some amino acid that's out in the protein, it's part of a loop or not particularly important, you may have plenty of structure in that protein for it to work effectively. If it's part though that's one of these bridges, or it's clearly a part of the amino acids that are critical for transmembrane connection of it, or where maybe one of the codons drives the production of an amino acid site where you've got a lipid deposition or something, and it's no longer there, well, the protein may not be able to be turned on or turned off, or it may not localize to the right part of the cell. And so it can have all sorts of effects, from everything from essentially no effect all the way again to where it's a completely dysfunctional protein. Okay. All right, well, those are the classics. All right, when we talk about transitions, transversions, we talk about silent, missense, nonsense, we talk about frame shift and in-frame. Those are the classics. That's what you would see in any genetics book. So we've gone through those. I want you to look at those, make sure you understand them. If you see, we might give you a code up here somewhere, a piece of DNA, make you take and transcribe it, and then say, oh, by the way, what happens if you change position number 16 to a G? What would happen at the end? And you say, oh, I go all the way down and tell me that it's ultimately going to be a missense or nonsense or whatever. Okay, so something that you could practice with a little bit. All right. Well, we have some more mutational things. We're going to up the scale a little bit and talk about mutations even more in the next couple of lecture sets.